All right, welcome to another week of Soul Seminar Online. Just a quick announcement. Hopefully, if you're in the UK or Europe, you were mindful of the time change and you're here. If not, I guess we'll see you when you sort this out. But just for a note, the US is having their daylight savings time ends this Sunday. So next week, we're going to be an hour earlier. Painful, I know, but we can do it. Today I want to introduce Elizabeth McCarthy. So Dr. McCarthy did her undergrad degree at Smith College. As an undergrad, she did her summer internship at the Royal Botanical Garden at Kew in London, where her relationship with Nicotiana began. By the end of the summer, she fell in love with Nicotiana, so she went back to work in London after graduating from Smith to complete her PhD jointly at Queen's Mary, University of London, the Royal Botanic Gardens, Kew, and the Natural History Museum. She did her postdoc at New York Botanical Garden and returned to studying Nicotiana during her postdoc at the University of California, Riverside. She's currently assistant professor in the biological science department at SUNY Cortland in Central New York. She is pursuing questions about flowering evolution and Nicotiana and several excellent undergrad researchers. And with that, you can share your screen, Elizabeth.
All right, let's get this rolling. Oops, wrong button, try again. All right, well, thank you so much, Chelsea, for the opportunity to talk today. I'm really excited to be here. So my name is Elizabeth McCarthy. My pronouns are she, her, hers. And today I'm speaking to you on the stolen land of the Haudenosaunee, the people who build the Longhouse, specifically the Onondaga and Tuscarora peoples. And so I'm excited to talk to you about the evolution of flower color in Nicotiana Allopolyploids today. And we're going to be moving from pigments to genes to modeling. And because, like Chelsea said, it's been a long time since I started working on Nicotiana. We're about, I think, at 16 years now, so time flies. And if anybody would like to email me, my email is here, or follow me on Twitter, I would love to chat with you. Okay, so today I'm going to take you through just an introduction to get us sorted, and then talk about 3 different pieces of a story about flower color evolution in Nicotiana polyploids, and then finish up with some current projects that my undergrads and I are working on. So polyploids involve whole genome duplication, and because they have these extra copies of genes, they're buffered from deleterious mutations. They tend to have higher tolerance to stressful conditions. They can colonize new niches or expand their ranges. And these functional divergence that can occur in these gene duplicates can lead to evolutionary novelty. And so I think they're really cool. And so that's one of the things that drew me to Nicotiana, since we have so many polyploids in the genus. So just to start off, what is ploidy? It's the number of sets of chromosomes that an individual has. So if you have 1 set, you're haploid. If you have 2 sets, you're diploid. And then 4 sets, you would be tetraploid. And so polyploid is having 3 or more sets of chromosomes. We can also have allopolyploidy, where we have whole genome duplication plus interspecific hybridization. So we can see that here with our 2 different flower colors and 2 different species represented in chromosomes with different colors. And then the allotetraploids will have 2 sets of a genome from each of their progenitors. And as you can see in the flowers, they can display phenotypes that are different from either of their progenitors. And so I'm interested in how this merger of different genomes back together in the same nucleus creates genomic shock, and how that genomic shock leads to differences in phenotype. So polyploidy has played a huge role in land plant evolution. Here on this phylogeny, all of the ancient polyploidy events are in red. And then in this violin plot, we can see that all of our land plants have at least 1 or maybe more, and we have lots in the angiosperms. So it's something that is occurring all of the time in the evolution of land plants. The other side of the story is that we have flower colors and morphologies that are going to be attracting different pollinators to visit them and spread pollen. And this is just a subset of some of those pollinators. And we can see that there's lots of different colors and shapes. And so this is kind of where my two pieces of my research program fit together: flower color evolution and how that affects pollinators, and the polyploidy. So many of the pigments that are going to produce different flower colors are anthocyanins or flavanols, and both of these are produced by the flavonoid biosynthetic pathway, which we can see here. So in this diagram, the bolded labels are the enzymes that are working together to create different pigments, and then we can see the pigments at the end of these arrows. So we have flavanols coming off here. These are colorless to humans, but absorb UV, and so can be have signals for pollinators who have UV receptors. And then down here we have colorful anthocyanins. And so these different pigments give off different colors. So pelargonidins are red, cyanidins are pink or magenta, sometimes blue, and then delphinidin are purple and blue pigments. And like I said about the flavanols, because they absorb UV, we can actually get different signals based on UV absorption or reflectance. So here in this petunia flower, there are lots of flavanols, and so they're absorbing UV light. In this petunia flower, there aren't as many flavanols, and so it reflects the UV light. So the overarching research questions of my program are: how does genome merger yield the novel and diverse phenotypes that we observe in allopolyploids? And then how do these phenotypes affect the evolution of allopolyploid species? So here is Nicotiana. And like I said, well, of course, it is my favorite, so we have to acknowledge that. It's my favorite because we have all of this diversity in floral form, and shape and size and color. And we also have a whole lot of allopolyploids. About half of the genus of Nicotiana are allopolyploids, and I'm going to introduce you to several of them. So Nicotiana aurentii, Nicotiana rustica, and Nicotiana tabacum are about 0.4 to 0.7 million years old. Section polydiclei has two species that diverge from a single event about 0.5 million years old. And section Repandae are four species that diverge from a single event about 4 million years old. And for the talk today, I'm going to be focusing on two of these, Nicotiana tabacum and section Repandae. So for Nicotiana tabacum, the diploid progenitors are Nicotiana sylvestris, which has a white flower, and Nicotiana tomentosiformis, which has a pink flower. Our tabacum accessions, which are about 0.6 million years old, are natural polyploids. We have variation in flower color and with different accessions. So we have a magenta flower, which is Nicotiana tabacum 095, or tab 095 for short during this talk. And we have a light pink one, Nicotiana tabacum chuvamani, which is going to be tab 2 for short in the talk. But we also have some synthetic polyploids that were created in the lab. And these are first generation polyploids, and they give us an idea of what's happening immediately following that polyploidization event from the same diploid progenitors that natural tabacum arose from. And again, we have some different flower colors here. QM24 has a lighter pink flower, QM25 has a darker pink flower. And so we can use these synthetic polyploids to compare to our natural polyploids to get an idea of how far they've come in the 0.6 million years that they've been in existence. So the first story that I want to share with you is about biochemistry and the pigments that are behind the different flower colors that we see. So the question here is, which pigments underlie these floral color differences? And this work was part of my postdoc work when I was working at the University of California, Riverside. In collaboration with Stacy Smith and Andrea Berardi, I got to visit Boulder and use the HPLC to answer this question about pigments that I had had since I did my PhD, so it was very exciting. And now that's been quite a while. But what we found was that the differences in these different pink hues in our Nicotiana tabacum polyploids polyploids and progenitors is mostly due to differences in cyanidin concentration. So here we have our different flowers on our x-axis. We have our diploid progenitors, our natural polyploids, and our synthetic polyploids. If we look at this graph up here, we have cyanidin, and you can see that our dark pink progenitor and our magenta polyploid have way more cyanidin than some of the others. The other interesting thing is when we look at pelargonidin, we see that there's a little bit of pelargonidin being produced by the natural polyploids that's not present in either of the diploid progenitors or in the synthetic polyploids. And so we can say that this is transgressive in these polyploids because it's not found in their diploid progenitors. It's not a whole lot of pigment, but it's there. And so I've always been curious about why it's happening. And so that's something that I would hopefully like to answer at some point in the future. We also measured the flavonol concentration. So here we have camphorol and quercetin. And so we can see that there's some variation in the camphorol and quercetin concentrations. Some of these accessions have more camphorol, some of them have more quercetin. So let's think about how this is actually being put together. Think about the flux of the pathway and how these pigments are being produced. So here we have our pathway again. If we look at our white parent, Nicotiana sylvestris, we know that we have low flavanols and no anthocyanins produced. So we can think about the flux of the pathway going in this direction towards our flavanols. In our pink progenitor, Nicotiana tomentosiformis, we've got high flavanols and high anthocyanins. So our flux through our pathway looks like this. If we're looking at our two different polyploids, our natural polyploids, the magenta in orange and the light pink in blue, we can see that we have variation. So one of them has low flavonoids and high anthocyanins. One of them has high flavonoids and low anthocyanins, but all of them are having this transgressive little bit of pelargonidin. So if we move on to the pigments in section Repandae, again, we have our species, oh, excuse me, I lost my mouse. Where did it go? Here it is. All right. Our species along the bottom, here's our diploid progenitor, again, Nicotiana sylvestris. Our paternal progenitor is Nicotiana obtusifolia. And then we have 3 species here that we're looking at, our polyploids. We have much higher camphorol in Nicotiana repandae and Stocktonii than either of their progenitors. We don't see very much difference in quercetin across any of these. And then really interestingly, we have transgressive delphinidin in Nicotiana and Repandae that we don't see in either of our progenitors. And so that's pretty exciting. We have this purple pigment from 2 white flowers coming together to create polyploids. Where is it coming from? So this is something that, again, I'm going to be investigating in future, not currently investigating now. So let's think about the flux through the pathway for these species. So again, our maternal progenitor has low flavonoids and no anthocyanins. Our paternal progenitor is similar. Stocktonii has a lot of flavanols, but no anthocyanins. And then both Nicotiana and Repandae have transgressive delphinidin, but Repandae has much more flavanols than Nicotiana does. And you can look at these and you're saying, well, those are white. Where is this delphinidin coming in? Where's the purple? So it's actually in the floral tube. So if you look at Nicotiana, there's this kind of blush of brownish purple pigment on the tube, which is where I think the delphinidin is coming from. And then in Nicotiana repandae, there's definitely purple on the inside of the tube. So that's where it is. Okay, so that was the pigment story. So from that, we can conclude that cyanidin concentration drives flower color differences in Nicotiana tabacum accessions, that our natural tabacum accessions have a little bit of transgressive pelargonidin being produced, that Nicotiana repandae and Stocktonii have higher camphorol levels than both of their progenitors, and that Nicotiana repandae and Nicotiana have transgressive delphinidin. So once we had the pigments, and we knew what was changing across our different flower colors, the next part is to ask questions about the genetic basis of those biochemical differences. And so we did some transcriptome analyses to get at that. But before I go into them, I'd like to talk about homolog expression bias. So if you have an allopolyploid, your progenitor copies are called homologs. And so just like we said before, if we have two different progenitors and their genomes represented in different colors, we're going to have two copies of each gene in our allotetraploids. So for example, the DFR gene, which is one of the genes that's going to be making anthocyanins in our pathway, will have a maternal copy here in purple and a paternal copy here in green. And if these are expressed at the same level, then there's no bias between the homologs. But if they're not, then we can say that there is a homolog expression bias. So here in this example, if we only have 2 copies being produced by the maternal copy versus 5 copies being produced by the paternal copy in terms of the transcripts, then we can say that it's going to be biased toward the paternal versus the maternal copy. And throughout the talk, I'm going to be reporting homolog expression bias as the percentage of the maternal homolog. Excuse me. So our questions here are, what is the genetic basis of the pigment differences that we observed? And do changes in homolog bias correlate with changes in flower color? So to do this, we did a developmental series where we picked 3 developmental stages of corolla tissue: early stage, where pigment was not being produced yet; a mid stage, where it had been; and a later stage right before enthesis. And this work still comes from my postdoc at the University of California, Riverside. And I worked with 2 fabulous undergraduates, Amber Lawhorn and Imelda Curti, on this portion of the project. So for our transcriptome analysis, we used corolla material for our 2 diploid progenitors, for our 2 natural tabacums of different flower colors, and our 2 synthetic lines that also have different flower colors. We chose these 3 developmental time points, like I pointed out earlier, and we did an Illumina platform with 1 by 85 base pair reads. We used those to create a de novo assembly, but unfortunately, because we had short reads, it was difficult to assemble and distinguish between our homologs because we just didn't have long enough data. And so to combat that, once I started at SUNY Cortland, we were able to add some long reads with Oxford Nanopore from both of our progenitors at the early and the late stage of development. We got 6.5 million reads from this data, and our read length N50 was just under 1 kb. And Jacob Landis was instrumental in this part of the project. And so we did a new assembly where we added the Illumina data, our long read Oxford Nanopore data, and data available from the ENA database. And we compared our 2 assemblies. So our Illumina only assemblies and our assemblies that had longer reads. And as you can see from this table, we had fewer contigs and longer N50s with the long read data. And if we look and compare our Busco gene analysis results, we had far fewer fragmented or missing genes with the addition of the long read data. And we were actually able to distinguish between our homologs. And so that is exactly what we're looking for. And so from these 2 progenitor assemblies, we made an in silico tabacum assembly by putting them together, and then used that to analyze the data. So in this graph, on the x-axis, we have different comparisons across our accessions. On the y-axis, we have log fold change, so how differentially expressed specific genes are. This is from our early developmental time point, and these violin plots are giving us an idea about all of the differentially expressed genes across the transcriptome. So violin plot is basically just like a histogram. And let me orient you to how we can interpret this. So for each of these comparisons, we have 1 species at the top and 1 at the bottom. And that's going to be correlated to the top and the bottom of the graph. So if a value was positive, that means that that gene was more highly expressed in Nicotiana sylvestris compared to Nicotiana tomentosiformis. If the value was negative, it was the reverse. It was more highly expressed in tomentosiformis versus sylvestris. So what I want you to take from this graph is an idea about how many differentially expressed genes we have, which is down here with the ends. So because we put together our 2 progenitors, into our assembly, we should see complete differential expression across all genes, because if we can distinguish between homologs, the sylvestrous ones are only going to map to the sylvestrous genome, the tomentosiformis ones are only going to map to the tomentosiformis genome. And we see that we have our highest proportion or number of differentially expressed genes in that comparison between our progenitors, over 43,000. Next, we're comparing our tabacum accessions to our maternal progenitor of Nicotiana sylvestris. And here we also expect to see higher levels of numbers of differentially expressed genes in our progenitors compared, the polyploids compared to our progenitors, because they have those tomentosiformis copies that the sylvestris genome does not. And that is what we see. But we see fewer genes differentially expressed than between our two progenitors. Next, we're comparing our polyploids to our paternal progenitor. We see similar trends like we saw with our maternal progenitor. And then our last three comparisons are within our polyploids. So between our synthetic polyploids and our natural polyploids, between our natural polyploids and between our synthetic polyploids. And what we would expect to see is far fewer differentially expressed genes here than the other comparisons we just talked about, which is what we see. We also expect to see more differentially expressed genes between our synthetic and natural because of the evolutionary time that the natural polyploids have had to diverge. Fewer between our two natural accessions, and then even fewer between our two synthetic accessions, which is what we see. So that was the entire transcriptome, but really we're interested in our anthocyanin genes and of our biosynthetic pathway. So I've given you a cheat sheet of the pathway. I've highlighted it in different colors, and so each of those colored arrows is going to correspond to the points to give you an idea of where in the pathway those genes are. The other thing we need to know is that our sylvestris copy, our maternal copy, is going to be transparent, and our paternal copy is going to be bold. And so that's what's going to allow us to distinguish between those. So again, along the x-axis, we have comparisons between different accessions, and our y-axis is looking at our log-fold change between expression. And we're going to have the violin plots that I showed you at the background. That's representing what we're seeing in the whole transcriptome. And then we're highlighting our anthocyanin genes with colorful points. So between our two progenitors, we can see that we have sylvestris genes more highly expressed in sylvestris, tomentosiformis genes more highly expressed in tomentosiformis, which is exactly what we want to be seeing, right? These should be mapping to different places in our in silico genome, and so we're seeing differential expression across. When we look at our tabacums compared to their maternal progenitor, that's white, we see that they have higher expression of tomentosiformis genes, but also you'll notice there are a few that have higher expression of sylvestris genes than are occurring in the sylvestris genome, which is pretty interesting. When we compare the polyploids to tomentosiformis, the pink parent, we see that sylvestris copies are more highly expressed in the polyploids as we expect, but we have some tomentosiformis genes that are actually more highly expressed in tomentosiformis than in any of the polyploids. When we start to look between our polyploids, we can see that we have far fewer differentially expressed genes. And what I want to draw your attention to are these purple dots. That represents DFR, which is that first gene on the way to making anthocyanins. And in each of our comparisons between dark pink and light pink flowers, we see that our DFR copies are more highly expressed in the dark pink compared to the light pink flower. So that's intriguing. So now we can ask the question of, do changes in homolog biomes correlate with flower color? So we're going to focus on DFR and the graphs that I'm going to show you in a bit. DFR again is at this position in our pathway towards making anthocyanins. We're going to be looking at data from the early developmental time point and the late developmental time point. We're going to be, our y-axis is going to give the percent of the sylvestris expression, and then the dotted line on the graphs is going to represent the progenitor ratio that we see when we compare sylvestris and tomentosiformis to each other. So on our x-axis, we have our different accessions, so our progenitor diploids, our natural polyploids, and our synthetic polyploids. And what we notice is that we do see some differences in homolog bias, but they don't seem to be correlated with differences in flower color. So our light pink flowers and our dark pink flowers, we don't see patterns there. We tend to see patterns between our natural polyploids compared to our synthetic polyploids instead. So it looks like homolog bias changes are not correlated with flower color. However, if we look at the overall expression of DFR, we do see that there is correlation with flower color. So these are similar graphs, but instead of the percentage of the homolog, we're looking at our summed TMM values, which represent transcript accumulation from our transcriptome analyses. And especially in early development, we start to see a pattern that correlates with flower color. So our dark pink progenitor, our magenta natural polyploid, and our dark pink synthetic polyploid have higher levels of DFR, but our white progenitor and our light pink ones are much lower. And actually, the light pink ones are nearly as low as the white progenitor early on in development. If we look later in development, we can see that changes. So our white parent stays very low, which makes sense because it doesn't produce any anthocyanins, but the accumulation of DFR transcript increases in all of our pink accessions. And so from this data, we can conclude that it's likely that DFR expression is delayed in our light pink flowers. And so it's not turned on early in development, it's turned on later in development. And so perhaps this is allowing for less cyanidin pigment to be produced, and that gives us our light pink phenotype. So another question we can ask with this data is, do we see common differentially expressed genes when we're looking at light versus dark pink comparisons within our polyploids? So we have 4 comparisons. Light pink individuals are going to be on this side, dark pink on this side. Again, we're going to be looking at different genes. So our cheat sheet is up here. And when I highlight those genes, they will pop up as colorful in the pathway. And so really interestingly, and we kind of saw this already on the violin plots, we see that all 4 of these allow us, are having higher expression of the tomentosiformis type DFR gene in the dark pink flowers compared to the light pink flowers. And that's why this is highlighted in magenta. And then if we look across the rest of this graph, we can see that some comparisons share different genes, and some have singleton genes that are differentially expressed, but some have higher levels of DFR from the sylvestris copy in our dark pink. Some are having higher flavonol synthase, which is the one that leads to flavanols up here in our light pink flowers compared to our dark pink flowers. And so we're seeing that at least the tomentosiformis DFR is consistent, and then we have some variation across others. So that's the end of the transcriptome story, and our conclusions there are that homolog bias does not correlate with differences in flower color in Nicotiana tabacum, but overall DFR transcript accumulation does. So it seems that DFR activation is delayed in light pink flowers, which allows less cyanidin to be produced and a light pink phenotype to come through, and that our tomentosiformis copy, which is the pink parent of DFR, is upregulated in our dark pink flowers compared to our light pink flowers in all comparisons between light and dark pink allopolyploids. Okay, so one more story, and this one is about modeling and using evolutionary simulations to answer some questions. So those questions are, what types of mutations are necessary to induce floral color shifts? And are some floral color shifts harder to evolve to than others? And then do the simulated enzyme concentrations predict what we actually observe in our transcript accumulation? So again, we're going to be talking about the flavonoid biosynthetic pathway here. These simulations are simulating the evolution of the genes within the pathway. However, we know that in our Nicotiana tabacum accessions, we don't actually produce any myristotin or delphinidin because they don't have a functional F3'5' gene. And so we truncated the pathway to make it a little bit more straightforward for our questions. And remember that the differences in the pink that we're seeing are mostly due to differences in cyanidin concentration. So this work, it would not be possible without Luke Wheeler and Stacy Smith. They published this paper putting together the evolutionary simulations that allow users to define a pigment optimum and then evolve the flavonoid biosynthetic pathway to produce that pigment optimum. So this work would not be possible without them. So thank you, Luke and Stacy. My research student, Lane Jensen, who has since graduated from SUNY Cortland and started a PhD program at the University of Buffalo, worked with me on this project. And so in the simulations, there are 3 enzyme parameters. So we have all of the enzymes in the pathway, and each of them is going to have an enzyme concentration, a kcat or catalytic constant, which determines how fast that reaction is going, and a km or the binding affinity of the enzyme for the substrates. And how the simulations progress is that a mutation is randomly introduced into one of these parameters for a single gene. And then if that mutation leads to a pigment composition that is closer to the user-defined optimum, it's kept and fixed. And then the next iteration moves on from there. And we run these simulations for 50,000 iterations or until the optimum has been reached. And for our work, we modified Luke and Stacy's simulation to be able to evolve to multiple optima, to be able to better represent the observed pigment data that I just told you about earlier. And Luke also gave us some help in figuring out how to tackle that problem. So thank you again, Luke. So we evolved to 3 optima. We chose Cyanidin, Pelargonidin, and Quercetin. And this was based on a percentage. So the overall output of the pathway stayed the same. And so the camphorol percentage was calculated automatically from our 3 optima. And those optima were based on the observed pigment compositions that we had for each of our 6 accessions, which we've talked about in detail already today. So the initial parameters of the pathway, we did some first simulations where we used those initial parameters and evolved towards our different pigment compositions for each of our accessions. And then we used those parameters as starting points to then ask questions about floral color shifts. And we did a lot of floral color shift simulations, but the ones I'm going to talk to you about today are synthetic polyploids to natural polyploids. And so asking the question of how did natural polyploids evolve different flower colors, using the synthetic polyploid as kind of the baseline of what we expect to see immediately following polyploidization. And then white to dark pink and dark pink to white transitions with using our progenitor diploids. Okay, so here we have all of our 6 accessions, which hopefully we're pretty feeling like we know them pretty well now. And each of these little cartoons gives the pathway, and the circles at the ends are representing the proportion of our different types of pigments. So for example, for tab 095, our magenta flower, we have more quercetin than we do camphorol, and we have a lot of cyanidin, compared to tab 2, where we have more camphorol, less quercetin, and far less cyanidin. The initial parameters in the pathway produce mainly camphorol and pelargonidin, and then secondarily quercetin and cyanidin. And so in those first simulations, like I talked about in the last slide, we use the initial parameters and evolved to each of our observed parameters for our accessions. And then we took those evolved parameters and use them as the starting point for our floral color shift ideas. So I have given you a cheat sheet up here for our pathway that's going to be hopefully easy to orient you when we're going through some of the next slides. So up here we have our flavanols, camphorol and quercetin, and our anthocyanins, pelargonidin and cyanidin. And when we're talking about specific genes in the pathway or enzymes in the pathway, I'm going to highlight those to give us an idea of where they are so we can all be on the same page. Okay, so our first questions are, how did natural accessions evolve different flower colors from the same polyploid origin? So to start off, we used the evolved parameters for our QM25 synthetic, and then we evolved towards a tabacum 095, our magenta natural polyploid, or our tab 2, our light pink polyploid. And these strip plots here are representing all of our different parameters. So we have our enzyme concentrations, our kcats, and our kms for all of the enzymes within our pathway. And the dots are going to be black if that parameter was unchanged throughout the simulation, or colorful if it was mutated to get to our user-defined optimum. And what we can notice just looking across all of this is that we see that there are mutations in all of these types of parameters, enzyme concentration, KCAT, and KM, but they're concentrated at just 3 genes, so F3'H, FLS, and DFR. And if we look up here at our cheat sheet, you can see that they're highlighted in red, and they're actually the ones that are at the branching points of this pathway. So it kind of makes sense to focus mutations at the branch points, because that's what's going to allow you to change the flux across the pathway, to have higher percentage of cyanidin or camphorol, depending on that user optimum. So if we dive in a little bit closer, here I've highlighted 2 parameters. The pink one is the KCAT of DFR working on DHQ, which is going to be this arrow in our pathway. And the gray is the KCAT for FLS working on DHK. So that's going to be this arrow in our pathway. And if we look at our 2 different types of simulations, moving towards magenta or towards light pink flowers, we can see that we've got reciprocal changes in these mutation values. So if we are moving towards a magenta flower, we are going to increase the efficiency of DFR working on DHQ. But if we're moving towards a light pink flower, we're going to decrease that efficiency. And then vice versa for FLS working on DHK. So we're seeing these reciprocal changes when we're moving in opposite directions, which makes a whole lot of sense. So if we compile all of the information from these graphs, and if there was an increased efficiency of the enzyme for the substrate, then it's going to be in a green arrow. If there is a decreased efficiency, it's going to be in a purple arrow. So what we can conclude is that we're going to have increased efficiency on the path leading to increased anthocyanin production in our simulations towards a magenta flower, and then a decrease in the efficiency in the enzymes that are moving towards flavonols. And then the complete opposite when we're simulating towards a light pink flower. So decreased efficiency in the path leading to cyanidin, increased efficiency in the path leading to flavonols. So like I said before, we're seeing that we are changing and having mutations in all 3 types of the parameters, the enzyme concentration, the KCAT, and the KM. But if we look at a correlation matrix, which is what we have here, you can see that you don't need to have mutations in multiple of these parameters for the same enzyme. One seems to do it, because I've highlighted those for F3'H here. So inside each of these boxes where these red lines align is giving us how often mutations are happening at the same time. And so you can see that each of these boxes has a pink color, and so that means that they're negatively correlated with each other. So it seems like if you mutate it in one parameter and do what it needs to do with the efficiency of the enzyme, then that's sufficient. We can also look at whether there are patterns in the steps and when these mutations occur. So in these graphs, we have the x-axis is the number of steps in the simulation. So step 1 is the first step, step 2 is the second, etc. And then on our x-axis, we have our different parameters, and then a measurement of how often those parameters are mutated at each step in purple. And so what we can see here, we have our QM25 to magenta simulations and our QM25 to light pink simulations over here. And for our magenta ones, we tend to first mutate either the DFR concentration or the parameters involving DHQ, versus in our evolving towards light pink, we tend to see differences occurring in the concentration of FLS and F3'H, or FLS DHK and F3'H DHK parameters. So we're seeing different things happening first in these 2 different types of our simulations. Excuse me. Okay, so now we're going to shift to one more question of what mutations are necessary to shift between white and dark pink flowers in our diploids. So here we have our white to dark pink simulations, and here we've got our dark pink to white simulations. We have similar strip plots like we had in our last ones, and we still have reciprocal mutations in the same parameters when we're moving in opposite directions. But you'll notice that we have far fewer simulations that have actually reached the optimum, especially for our white to dark pink simulation. So we only had 10 that actually went to the optimum, and that became a 0.4% success rate for the white to dark pink compared to a 31% success rate for dark pink to white simulations. So that suggests that it's actually harder to evolve from a white flower to a dark pink flower than vice versa. And so we delved into this a little bit more by taking all of our comparisons and compiling whether we were evolving towards a higher level of cyanidin, towards a lower level of cyanidin, or towards similar levels. And so we compared how many steps it took to reach the optimum to answer the question of, does evolving towards a higher cyanidin level require more evolutionary steps? So we have our different comparisons, our evolution of cyanidin on the x-axis. So here we've got similar levels of cyanidin to a higher level of cyanidin or to a lower level of cyanidin. And on our y-axis, we have how many fixed mutations it took to reach the optimum. And from this data, what we see is that yes, it does take more steps to evolve towards higher cyanidin levels than it does to a lower level or between similar levels. So that's kind of exciting, that it does take more to get to a higher level of cyanidin. The last thing that we looked at for these simulations is we compared the enzyme concentration parameters to the actual observed transcript accumulation levels from our transcriptome analysis. And so in this graph, we have our enzyme concentrations from the simulations on the x-axis and the transcript data on the y-axis. And we looked at only the genes that actually changed in our simulations. So we narrowed that down to DFR, F3'H, and FLS. And we did find that there is significant correlation between these two different sets of data. However, only about a third of that data, or a third of the correlation is explained by what we see here, or by this relationship. But still cool that we're seeing that our simulations are actually giving similar results to what we see in our transcript levels from our transcriptome analyses. So to conclude what we know from our simulations, it seems that the branch point enzymes of F3'H, FLS, and DFR are driving the evolution of pigment changes because we see mutations in all of these, but also in all three of the parameter types that are observed: the enzyme concentration, the KCAT, and the KM. We saw reciprocal mutations in simulations where we have opposite directions of the same floral color shift. We see that evolution towards a higher level of cyanidin requires more steps than evolving to a lower level of cyanidin, and our simulated enzyme concentrations seem to predict our observed transcript levels. So to conclude the talk today, I'm going to give you a little taste of what we're doing in my lab right now. So my undergraduate student, Abigail McCoy, is looking at the question of, does positive selection correlate with floral pigment shifts? And so we're doing DNDS analyses on our flavonoid biosynthetic pathway genes to determine if there's positive selection or even just relaxed selection on pigment shift branches. And we're looking at pigment shifts either just anthocyanins versus no anthocyanins, or looking at differences between anthocyanins, because we have some Nicotiana species that have cyanidin anthocyanins and some that have, delphinidin. And so here we've got some trees. We're going to be using available data from
Oh no, I think Elizabeth was muted. I don't know.
Yeah, it looks like she's frozen on my end.
Okay. I was like, is this my computer? Okay. we'll see if she rejoins. Maybe people can think about if they have questions for Elizabeth because she's wrapping up. If not, maybe it'll work out.
Yeah, maybe we'll give it a second. It could be a
You're back. Good.
Wi-Fi issue.
Oh, sorry about that. My Wi-Fi likes to just drop in my office. Let me get back to where we need to be. Sorry about that, folks. Okay. Everybody can see that? All right, let's start this one again. Trinity Tobin is going to be asking the question of, do differences in biased homolog expression correlate with changes in flower color in the Repandae species? So we're going to be doing some droplet digital PCR on flavonoid biosynthetic genes to get an idea about whether homolog biases are changing. Here we didn't see that they are affecting or correlating with the changes in tabacum, but maybe it'll be a different story in Repandae. And to conclude, I would just like to thank with some end with some acknowledgements. So this has been, a very long story, and very most of my research career. And so I'd like to thank my 3 PhD supervisors, Andrew Leach, Mark Chase, and Sandy Knapp, my postdoc supervisor, Amy Litt, collaborators from my PhD, collaborators from my postdoc. I would like to especially highlight Jacob Landis for his work on the nanopore data, Stacy Smith, Andrea Berardi, and Luke Wheeler for their help with pigments and simulations, and then my fabulous undergraduates, Imelda Cardi, Amber Lawhorn, Lane Jensen, Abby McCoy, and Trinity Tobin. And with that, we have to have jazz hands. There can't not be jazz hands. And so we even made them happen in a pandemic. Both Abby and Trinity just presented some posters based on their work at an undergrad symposium that we had the other month. And so I wanted to highlight that as well. So thanks so much. And I will take any questions that you have.
Thank you so much, Elizabeth. So we'll take questions now. So you can either type them out if you have them. You can just type questions and we can kind of go from there if people have questions. Awesome research. I think it's great that you can type polyploidy to floral color evolution, which is probably no easy feat. So it's really cool that you're able to do that.
Hi, Stephen.
So
hey, can I ask a question or just
Please.
a comment? Thank you. That was super interesting. Yeah, I read some of those recent papers, but it's really nice to hear your talk on that. I just couldn't help but think that we should work together on some of these suavilentis and look at what's going on.
Yes.
Because they're white. Everyone's thing is, oh, they're just white flowers, but they're not actually all the same kind of white. And I kept thinking there must be flavonol differences.
Mm-hmm.
Also, some of them you do get that flushing on the corolla tube as well. So I wonder whether there's something going on there.
You sent me a picture one time from the field where it was like super purple.
Yeah.
So yes, I want to know the answer to those questions. Let's do that.
Because there's also, for me also, I'm interested in like the flower size and the shape difference, and some of them are not obligately selfing. So so there's a whole bunch of stuff going on there. But this was super interesting for me as well to think actually they're not just white. There are these major differences that either maybe it's on somewhere else on the flower or maybe it's about the UV absorbance. So yeah, let's do it. It's not so simple from the parental point of view, maybe because of the issue with a super complicated maternal progenitor, but we'll forget that. We've got so many species now as well. So
yeah, and that's what I'm still kind of waiting to hear about, like how many suavilentis species do we actually have now? I don't know.
It just keeps going up.
We thought we had when I did my PhD. So yeah, but I would definitely be interested in tackling those species at some point. So let's be in touch.
Yeah, definitely. And thanks again. Really, really super interesting stuff.
Thank you for coming. It's great to see you.
Awesome. Are there any other questions that anybody has? How hard is it to make a new polyploid?
So I didn't actually do those experiments. That was postdoc in my postdoc lab who worked on, my PhD lab who worked on those. They actually crossed autotetraploids of the diploid progenitors and then hybridized them. That seemed to work better than trying to do colchicine induction of the diploid hybrids. So I don't have the experience. One of the things that we're still trying to figure out is that those synthetic hybrids are still first generation because they don't really set seed very well. And so I don't have any second generation ones. But I think, and maybe Stephen knows more about this, but I think that some folks working on tabacum in France have some newer synthetics, which I should talk to them about because it would be cool to see what they're doing there.
Yeah, I think so. Those would be interesting, definitely.
Hester, you have a question?
Yeah, hi Elizabeth, thanks. Really nice talk. Yeah, nice to see you. I'm just wondering really if you're going to try and investigate any of the kind of hypotheses that the modeling generated but in wet lab work. So like, I don't know, looking at enzyme activity and that kind of thing.
Yes.
I think I also have to say that those graphs were amazing, but I'm going to have to like stare at them for another hour or two, I think, to try and understand all of it.
Stuff. So thank you. Yeah, so that's definitely something that I'm looking forward to doing. So I have a chemistry collaborator here at SUNY Cortland who does enzyme work. And so we're hoping to collaborate and do some of the enzyme assays to see whether what we predict from the simulations are actually happening with our definitely DFR. And I know that lots of people done DFR assays. I would love to do F' H and FLS at some point too. We'll see how that goes. I don't know if those, I think they, or at least FLS has been done, but not as often. And so we'll see. We'll see how well it goes. We'll start small, get bigger. But yes, and Trinity Tobin, my undergraduate who just started working with me, has a biochemical interest. And so hopefully she's going to pick up on that project too.
Cool, that'll be great to see. And I might have missed it. Have you published this work already, or is it?
Some of it's published, the simulation stuff not yet.
Okay, cool. Great, thanks.
But I'm excited also because both Abby and Trinity are just sophomores, and so I'm hoping that I get to work with them for a lot longer.
Awesome. Is there any last questions for Elizabeth? I don't see anything. So if you have questions in the future, you can definitely email Elizabeth. She provided her email.
My email in the chat here.
Yeah. Or follow her on Twitter, or she's definitely available. I'm willing to talk. So I just want to introduce next week's speaker. So you guys come and join us next week. We're going to be talking about integration, breeding, and eggplants from Jamie Prohens. So definitely tune in next week. And thanks again, Elizabeth, for coming and sharing your research with us. It's great.
Thank you very much for having me.
All right. Bye, guys.